My name is Kaylee Tallon, and I will be giving a literary review presentation on Pterois volitans, salinity tolerance, and effects. Here is the background information. Here is the picture on the left depicts a body plan of a lionfish showing the dimensions about how big it is, and then the image on the right shows the anatomical features of the lionfish, including colorations, number of spines and rays, dorsal and anal, and showing where the venomous spines are. Lionfish typically live in coral reef habitats and seagrass habitats. They have a rapid year-round reproduction with reproducing a lot of eggs at once. Their diet consists of a lot of smaller fish, crustaceans, bivalves, and cephalopods like squid. Their behavior is shown to have a higher salinity tolerance, generalist eaters, predatory at night, and venomous spines. Here is the global distribution of two different species of lionfish. P. volitans is depicted in the red as their non-native range, and then their native range for P. volitans is in the green in the Pacific Ocean, and for P. miles in the blue in the Indian Ocean. Here is the review work. The study purpose is to examine the salinity tolerance of lionfish and provide information for management strategies that could mitigate lionfish invasions in variable salinity environments. As salinity significantly influences the ability of aquatic organisms to distribute and move to new locations, lionfish are found to have a higher salinity tolerance as they're being found in brackish water systems, and brackish water ecosystems are highly vulnerable with highly vulnerable species as they are used as nesting and reproducing habitats. There was 3 different methods that I examined in my research: field observations, lab experiments, and environmental DNA. For field observations, they tested the use of mangroves by lionfish in the Bahamas, and they used hand nets while snorkeling to find the lionfish and observe them, and they collected DNA samples of the lionfish species. They also did a habitat use comparison, also in the Bahamas, using a spear gun while snorkeling to capture them, and they tested the stomach content, body size, stable isotopes, and conducted a CPUE. A third was the Florida estuary invasion in a river in Florida, and they used hand net sampling while snorkeling to observe where the lionfish were. And then there was an attempt to kill all lionfish via pole spears, and they measured length and did stomach content analysis to see what they were eating in the estuary. For lab experiments, they tested physiological limits in Key West, Florida. They collected different things to test their respiratory parameters, and they were kept in recirculating tanks to see where lionfish could survive based on salinity. In the Keys in North Carolina coast, they tested survival and growth of lionfish and collected temperature, pH, DO, and salinity, and they took measurements of total and standard length and mass based on the salinity control tanks. And then also in another river in Florida, they tested the salinity tolerance of lionfish, and they were caught in mesh cages when they found them, and then hand nets during the lab. And they were found after heavy precipitation in these lower salinity environments. They collected weight, length, and behavior observations based on different salinity tanks. They also tested effects and impacts in lower salinity in the Bahamas, and they caught them scuba diving using plastic hand nets, collected total standard length, weight, external muscle tagging, metabolic rates, and specific dynamic action in salinity tanks. Then there was a behavioral species interaction experiment where they tested the behavior of lionfish and snapper in different salinity tanks. They collected standard and mean lengths, used video analysis, and a behavioral ethogram to test how these species interacted depending on the salinity. Then one experiment was testing and found negative biological effects. That was also in the Bahamas. They took weight, length, external tags, O2 consumption, and metabolic rates. And they was to study the metabolic rate, food consumption, digestion, and growth based on salinity measurements. Then one experiment tested and used eDNA in 4 rivers and 2 bays of the Gulf of Mexico. They collected the eDNA samples in the water samples. They extracted that DNA and used polymerase chain reaction to see what exactly species were found there. The environmental DNA assay developed to identify the presence of lionfish DNA in the northern Gulf of Mexico, and the map on the left shows the different sample sites in the Gulf. With those 3 types of methods, we have the results. This result was from the Florida estuary invasion, showing the size distribution of 145 lionfish that were captured between different months. Which those results showed successful recruitment could have occurred many times throughout 2010 and early 2011, and that adaptations of lionfish would to have occurred for their presence in lower salinity environments, and that lionfish could have used structures as refuge in estuaries such as mangroves, and size could be dependent on high salinity or in low seasons. This graph was from the testing of survival and growth of lionfish, which shows the mass of lionfish during salinity experiments. And each colored line showed the different treatments of salinity. And as you can see, there's not a huge difference between the salinity effects and the mass of lionfish, meaning that their growth wouldn't be changed. As lionfish mass did not differ, as our ANOVA result was 0.56 across all treatments, and that showed lionfish are capable of tolerating lower salinities, at least in the short-term environments. But this further inducing their ability to inhabit lower salinity environments if they can still grow properly. Then these graphs were from the behavioral species interactions between the lionfish and red snapper. They were placed in different salinity tanks, and both species were placed in those tanks to see how they interacted with each other. And the chart depicts the different types of behavior and the description specifically of which species did what, and then if it was aggressive or based on fear. And they did all sorts of behavioral analysis, doing chases, charges, fin displays, displacements, avoidance, stalking, and standoffs between these 2 species. And then these results showed that lionfish displayed a lower aggression and predatory behavior in lower salinity environments, while snappers showed different patterns of avoidance and aggression depending on the salinity levels, which just shows environmental conditions influence behavioral interactions between these species, which could also mean different behavioral interactions between the native species in the estuaries. And then this graph shows the negative biological effects that came from the salinity in lab treatments. The first section was growth versus mass based on salinity. And as you can see, that we have a negative relationship. And then the bottom left graph shows metabolic and food consumption based on the lionfish's weight over time through the experiment, and then the big graph on the right shows digestion parameters based on weight. And then this showed that exposure to low salinity conditions reduced lionfish's metabolic rate, their food consumption, and overall growth, which suggested that survivability in low salinity environments decreases the lionfish's performance. Although it's shown that lionfish have adjusted to diverse environmental conditions, it was assumed that their invasion would be less successful in areas where the salinity is always low, as they move slower, they can't digest things as quickly, they don't eat as much, and their growth is pretty much slowed down, making them less of an issue as they are in other habitats. And then this result shows the eDNA experiment for lionfish detection. We have the different bays in different areas that were tested between October and July. Each colored bar shows the different areas that were tested, and depending on how high up the graph, it shows how positive detections of lionfish for all locations were being shown. And then this experiment clearly showed that there was a presence of lionfish genes within those water samples, which also suggests further invasion into newer habitats such as estuaries and lower salinity environments. And this experiment was used to show that eDNA can be used as an early detection method for invasive species that's non-invasive for the habitat and the ecosystem, as you just take a water sample and test if the DNA of that species is in there. And in conclusion, lionfish have shown to have an ability to tolerate lower salinity environments, and that can contribute to their invasion and expansion into estuary and other coastal habitats. But their invasion threatens native species, disrupts ecosystems, complicates management efforts, and just changes the whole dynamic food web behavior between different species. Changes in salinity impact predation risk, aggression, changes in habitat association, such as mangrove over coral reef, or vice versa, just showing which habitat lionfish prefer, and overall changes in behavioral dynamics based on the salinity levels. And that eDNA can be used as an early detection management for invasive species. But overall, there needs to be a targeted management system specific for these species, as traditional marine-based control measures may be insufficient due to the characteristics of the lionfish, as they reproduce extremely quickly, they eat about anything, they have no natural predators. But combining public awareness, incentives for harvest, habitat preservation, active removal efforts, and the use of salinity as potential control factor will be vital in developing successful mitigation programs and starting the removal of invasive species at a more efficient rate, especially now that they're being seen present in estuaries and other coastal ecosystems with really vulnerable species and habitats. And all of my references were provided in the slides throughout all pictures and diagrams. Thank you.